Hi guys, and welcome to the Chapter 13.4 video of the AP Bio Exam Review series. This is just on biotechnology, so the rest of Chapter 13 was in Unit 3, I think, or maybe Unit 2, but for our purposes, this is just going to be about that one section on biotechnology. Okay, you need to know the terminology of biotech. So we're going to just go over one figure real quick, but then also just really pay attention during the video to the words that I use and my word choice. That will help you know the terminology. There's nothing altogether too fancy about it, but you just need to make sure that you're using these words properly. So again, for the purposes of understanding the terminology, I'm going to go through figure 13.23, and this is one of the images from the book that was about using a restriction enzyme and DNA ligase to make recombinant DNA. And really just the title of this gets you most of the terminology that you need. Restriction enzyme is the first word. Restriction enzymes cut DNA at what we call a restriction site. Restriction site. Those are, like we talked about in class, usually the palindromes, the things that are the same way forwards and backwards. And so you can see in this particular diagram that, where's my little laser pointer thing? I think it's this one. That's not it. Here it is. That we have CTTAAG this way, and then CTTAAG this way. So that makes it the palindrome. It's the same way going this way as going this way. That's usually what restriction sites are, and they're these short little bands of DNA that an enzyme cuts it in a way, like here between the C and the T, or I'm sorry, the G and the A right here, and the G and the A right here, so that it's staggered, the ends are staggered. That causes this to have what's called a sticky end, and a sticky end is where there's just one of the strands of DNA are exposed, rather than both. When there's two strands together, then that's a stable molecule. But when we have this part that projects here, we call it a sticky end because that's not a stable molecule. So if we use a restriction enzyme, that restriction enzyme is going to go ahead and cut the covalent bonds, and that's what it's done here, and then the hydrogen bonds kind of fall apart that are in between them. If you use the same restriction enzyme on multiple DNA molecules, you end up with complementary ends. And that's what this is showing you. It's showing you that we have used the same restriction enzyme on this piece of DNA as well as with this piece of DNA. So then, once we've cut our DNA using these restriction enzymes, we throw all of these little chunks together in the same test tube, and then we hope that they pair up like this. Now we know they're going to pair up somehow because they have these sticky ends and that's really unstable. But what we're hoping for is that we end up with this. And that's why it says right here, one possible combination. Because the reality is we have absolutely no control over how these sticky ends reassemble. So even though we took a restriction enzyme to cut this, it's possible that it's just going to go back together in that way. Similarly with this other piece of DNA. This could just form a little circular plasmid or something like that. But we're going to hope that at least sometimes those sticky ends go together in our desired arrangement. But we have to then reform the covalent bond that we broke using those restriction enzymes. You can see that it's broken because of the little gaps here and here. And so we use DNA ligase to go ahead and seal those strands together. So now we have one piece of DNA with two strands that are consistent. They have the sugar phosphate band all the way down their 5' to 3' ends. And we call that a recombinant DNA molecule because it has DNA from two different sources in it. So, that's the summary of the whole process of making recombinant DNA. We'll look at this again a little bit later, but for now, just understanding the vocab is really, really useful. Okay, you have to know how plasmids are used to clone genes. So here's the diagram from your book that was about gene cloning and its uses, and I'm going to remind you one more time that when you hear the word cloning, we're not talking about Dolly the sheep. That's not what we're talking about. You want to remember that for all of our bacteria, all of our prokaryotes, when they go through this process of reproduction, they just bud. They go through this process of forming clones of themselves because it's asexual reproduction. They go through a process of forming another cell that has the exact same DNA as itself. And so because of that, when we think about cloning, we need to think about bacteria just producing colonies of all of the same organism. And so what this is showing you is that we have our bacteria, and we have a eukaryote, and we know that because we have a nucleus there. And so we're interested in one particular gene right here. And so we used the process of before on this last slide of performing, using restriction enzymes to make a recombinant plasmid. And so once we do that, then we can take that plasmid and insert it into a bacteria. So you can see how here we had an unrecombined plasmid, and then we recombined the plasmid, and then we put it back in the bacteria. Now, anytime this cell divides, it's going to divide with that gene. So then, well, what do we do with that gene? Well, we have to get the bacteria transcribe and translate it into whatever the target protein is. And so over on this branch, we're talking about, okay, we're going to use these bacteria, we're going to stimulate them to transcribe, translate, produce a protein, and then we're going to use that protein for our own personal uses. Many drugs are made this way. A lot of medical uses are taken care of in this way. Insulin is produced this way. So we use protein that is harvested because we tricked a bacteria into making it for us. We can also use this same procedure to just make many, many copies of a gene. And so if we take this bacteria and we allow him to reproduce and make clones of himself, he's actually also going to make clones of that gene of interest. Then we can chop that gene of interest out and insert it into other organisms in order to modify them in some way. And so this is just a way for us to make lots and lots and lots of copies of that gene in a natural way. You need to know the key ideas that make PCR possible and applications of this technology. So all of the techniques of biotechnology all focus around one feature of DNA that is of huge importance. And that feature of DNA is that the two strands are complementary, which means all of the information for both strands is really held in just one, because all it has to do is make the complementary copy in order to have all of that information. PCR is no different. And so with PCR, we have this three-step process of denaturing, annealing, and extending. And so denaturing is literally pulling apart your DNA strands. We break those hydrogen bonds that are holding those two strands together. Annealing is when we're adding the primers. The primers are introduced by a scientist, and so the primers aren't random. But what happens is if the scientist knows that they're trying to target this particular blue gene here, then what they're going to do is put primers in that are the ends of that gene, and they put that into the test tube when they go through this process. That way, these primers are going to stick only where they are supposed to. And once we have the primers attached, then extending is really easy. Because even in the process of PCR, we can't break that fundamental rule that DNA can't start replicating by itself, that one DNA nucleotide has to have something else to attach to. And so the reason we put those primers in is to tell the DNA, this is where I need you to copy. You're going to notice that this is copying and extending all the way, and it doesn't stop when it gets to the end of the gene. It keeps going and keeps going and keeps going and keeps going. Well, that's okay, but that's not what we're actually trying to do, because the goal is to just make this small blue section. And so we ended up making more than we needed to. But by going through multiple cycles of this, every time we make 2 more molecules, by the time we get to our 3rd cycle, you can see that we're making DNA strands that are just the DNA target sequence. We don't have any of the gray stuff going on either sides anymore. And so that it takes us 3 cycles to get to that point, but at some point we do get just the strand of interest. This is a very fast process. Scientists can put this in what we call a thermocycler, and it will happen overnight and make literally thousands of copies of DNA. The trouble is, even though it's extremely accurate, it's not as accurate as life. And the reason for that is because we don't have any of the enzymes that go through the proofreading and any of that kind of stuff in this process. And so when DNA is replicating in cells, there are dozens of enzymes that go through the cell after the DNA replication and make sure that the DNA is replicated properly. Those enzymes don't exist in PCR. And so because those enzymes don't exist in PCR, there is a limit to how many replications are useful within PCR, just because every time we do this, we are introducing small but significant errors into those DNA sequences. You have to know how gel electrophoresis can be used to separate DNA fragments or protein molecules. All right, so here's kind of the summary diagram from gel electrophoresis. And in this diagram, it's important that you remember that we have a negative side and a positive side, and our DNA goes right here. But DNA is negatively charged, and so it's going to move toward the positive end of the gel. The larger the molecules are, the slower they move. So here I can see the wells are over here. This is going to be the largest molecule on here, and these are going to be the smallest molecules on here. And the reason is because those shorter molecules are less impeded than the larger ones as they travel through this gel. And you guys saw an animation of that. Usually when you do this, you use what's called a ladder or a size standard on one side so that you can then compare your data, which is probably going to be this part over here. You compare your data to your size standard and you know how large those molecules are. Ignore this blank slide too. Sorry, I'm inept. Oh wait, maybe I can just delete it. Can I delete it? Okay, so once you've run your gel electrophoresis, you have these results, and so we're going to look at some sample results to give you an idea for what you should look at. So here's an example of PCR results. This is actually fairly realistic because here you can see that, the bands are really actually pretty hard to see apart. And on here we have like suspects, and then we have a blood stain, and the question is which one of these suspects was present at the scene. Of course, this doesn't mean that that person did it. It just means that the person was present. And so when you're looking at something like this, all you're doing is you're trying to figure out, okay, this is already a blood stain. We just match up our bands and find that right here, suspect number 3 is the only one who has the exact same fingerprint as everybody else. The question, we have 2 questions here. We're going to start with the second one. What do the thick versus the thin bands mean? I told you earlier that the further the band travels, the smaller it is. But the thicker ones, such as this is a pretty thick one versus the thinner ones, that's just telling you the quantity of DNA that is present there. And so how many molecules are that long that would accumulate in that spot in the gene, or in the gel rather. So that's what the thick versus the thin means. The question is, what gives everybody a DNA fingerprint? We talk about how DNA is unique to all humans, but then we also talk about how there's a significant amount of conservation between individuals that really, as far as genes that code for proteins are concerned, there's really very little variation. Well, the issue is actually in the introns. Introns have an awful lot of variation, and so it's variation in the introns that cause this to happen. And so when you're making a gel, what you do is you take the whole DNA from an organism, a human in this case, and you cut all of the DNA with the same restriction enzymes, and that takes, your 46 chromosomes, or if you're dealing with something different, the however many chromosomes there are in that organism, and it breaks them into these smaller chunks. And then those smaller chunks are run on a gel, and then it's cutting them usually in those introns, not in the exons. And that's what's giving everybody a unique DNA fingerprint. We also have some silent mutations in our genes that make us different, but that don't actually change the protein that's produced. And that's another reason why we have some unique, a unique DNA fingerprint when we don't make different proteins. But the great majority of the uniqueness for everybody's DNA fingerprint actually is coming from differences in those introns. So here's one where we're not actually looking for an exact match. Here where we're looking to figure out who's the mother and we know who the mother is, who's the father of this child. So if C is your child and here's mom, you know that the child has half the chromosomes from mom and half the chromosomes from dad. And so in this case, you look at the child and you say, okay, this band came from mom, this band didn't come from mom, so it would have had to come from dad, so there it is on the dad side. Father 2 in this case is the father of that child. So this is something we didn't do in class, but I'm going to go ahead and put it on here just in case you see it on your test, so that you don't flip out. I talked about using a size standard or a ladder to understand how long your DNA chunks actually are. And usually this is done on a log scale just because it's not a linear scale length to travel distance. And so what you end up doing is you end up saying, okay, well, this is how far I migrated, and that goes on your x-axis, and this is the molecular weight. And you make a standard curve based on that ladder. And so when you buy a ladder, it tells you this test tube contains DNA segments that are, 10, 20, 30, 40, 50, 60, 70 base pairs long. And so then if you know that when you get your gel out, you can say, okay, well, in this ladder tube, I know that this shortest strand was 10 base pairs long, and I know that it moved, let's see, molecular or migration distance, it moved this far. And so you're able to plot that, and then using a ruler, you can say, okay, well, how far did my unknown DNA move? And you're able to use a standard curve to figure that out. So it's usually done on a log scale, again, because it's just not a linear relationship, although you could do it on regular graph paper. It would work the same. But usually what we see is on this semi-log paper. So we'll see log on the y-axis, but not on the x-axis. So that is everything you need to know about biotechnology. I hope you found it useful. Good luck on your quiz, and I'll see you later.